Hello, I hope you're enjoying your day so far. Our project is investigating the relationship between mass and contraction velocity of cardiac myosin protein in birds. Over 9,600 species of birds have been discovered, yet cardiac muscle proteins have been studied in only a tiny fraction of these species. We are all aware to the extent to which cardiovascular diseases in humans impact upon individuals and society. Research into human and mammalian cardiac muscle proteins has led to advances in treatment and diagnosis of heart disease. Using human tissue is clearly not appropriate for a school-based project. Birds have higher metabolic requirements for flight and possess a similar four-chambered heart to humans. Birds too suffer from cardiovascular diseases. In farmed poultry, these pose a significant risk to the animals. Cardiac myosin isoforms in birds are little studied, and our project aimed to mirror the types of studies being carried out in humans and mammals to give us better understanding of the process and techniques involved and to investigate a little-known area. We aim to identify differences between the cardiac muscle proteins of a variety of bird species. There is a well-known relationship between the body mass of mammals and their heart rate. The larger the animal, the lower the heart rate. This inverse correlation also holds true for bird species. As you can see from this graph, for smaller animals with a faster heart rate, there is a corresponding reduction in the time taken for one cardiac cycle to be completed. The reduction in length of one cardiac cycle has been found in mammals to be due to cardiac tissue itself, which is largely composed of the protein myosin. This contracts at a lower velocity in larger animals. How is this brought about? What is it about the myosin that enables it to contract at such different speeds?
We began with research into muscle function and myosin proteins. Myosins are a large family of ATP-driven proteins that use actin as a track along which to move. Genomic studies have revealed 35 classes of myosin as shown in this phylogenetic tree. The group of myosin shown in blue in the top left of the tree include those found in cardiac tissue. Isoforms are proteins very similar to each other and able to replace each other. Slight diversity in their amino acid sequences give rise to different structural and functional properties. Striated muscles use different isoforms of myosin 2 subgroup. Most mammals express up to a dozen myosin 2 isoforms. These isoforms are named according to the gene coding for the protein. MYH6 and MYH7 code for the two cardiac forms alpha and beta myosin in humans. Myosin 2 isoforms contain two heavy chains, or MHCs, each about 2,000 amino acids in length, which constitute the head and tail domains. 4 myosin-like chains, MLCs, 2 per head, bind the heavy chains. In muscle cells, the long coiled tails of the individual myosin molecules join, forming the thick filaments of a sarcomere. The force-producing head domains stick out from the sides of the thick filament, ready to walk along the adjacent actin-based thin filaments. Velocity has been found to be a characteristic of the isoform, and the same isoform gives a slower maximum velocity from muscles of larger animals compared with small animals. The assumption is that small sequences change between isoforms and between species are responsible for these differences. So where should we be looking for these differences? Studies have shown that for embryonic and non-muscle myosins that the tail regions show significant divergence whilst head regions are well conserved. For beta cardiac myosin, the protein is true, which suggests that it is the head region which might account for the differences in contraction velocity.
The MYHC gene family in birds is more complex than in mammals. There are greater numbers of MYHC genes and isoforms in avian mussels. In mammals, developmental isoforms are usually restricted to early stages of development, whereas in birds, the developmental isoforms are also expressed in adults. Initially, we need to focus on extracting the myosin from a range of bird species and measuring the contraction velocity of the extracted myosin. For the protein to be of high enough quality, the bird hearts must be frozen a short time after death. The university has provided samples of crow, quail and chicken. We have sourced pheasants and ostrich. The picture shows us with the ostrich heart, which weighed in at over 1 kilo. We have made links with the Worshipful Company of Poachers, who have put us in touch with farmers keen to support the project, so we'll have some further chicken and turkey material shortly. Small samples of the muscle tissue are homogenised to break up the muscle fibres. As homogenisers are very expensive, we tried the slightly less sophisticated technique of hammering the samples on ice blocks and found this to be sufficiently effective. During the extraction process, the samples need to remain on ice as much as possible to preserve the protein samples. The quantities are very small, and it has not always been easy to identify whether we still have material present. Electrophoresis was carried out on the extracted myosin samples, run against a comparison of pure myosin to check that the myosin extraction had been successful. The protein samples were boiled in a sample buffer and loaded into the wells in the gel plates ready for electrophoresis. Our early gels showed no brands, and the university ran the samples again. These results showed that myosin extraction had been successful, with clear bands visible corresponding to the myosin heavy and light chains.
For the following sessions, unfortunately we had issues with the running of our gels, so we were unable to gain usable results. The University of Kent provided us some samples of crow and quail to run alongside the pheasant. For this session, we adjusted the myosin extraction protocol to speed it up so that we could run the extraction and the gels within the time available in the school day. The results were positive in that we could see bands for all the three birds, although the pheasant sample needed to be precipitated and run again to gain a more distinct band. The ostrich results also looked promising. We felt confident that we could now successfully extract myosin, so we were ready for the next phase. We took our samples to the university labs and carried out some tests there. The gel bands are cut out and digested using trypsin. The gel is then extracted with a buffer to draw the fragments out. The separated fragments of different sizes are injected into the mass spectrometer. By comparing the mass of the fragments with those of the known myosin peptides, we can evaluate if isoforms of myosin are present. Our results for the crow and quail match myosin 7 peptides from other species. We are awaiting the results for the ostrich. We also used our samples to measure the contraction velocity. This can be measured using an in vitro motility assay. The myosin sample is placed on a coated glass slide and actin is added. The myosin sticks to the slide and actin attaches the myosin to the myosin. The actin is then labelled with a fluorescent dye so that when the myosin pulls the actin chain, the movement of the actin can be viewed with a fluorescent microscope. The distance travelled by the actin and time taken are recorded, and the contraction velocity can be calculated. Our samples did not produce images of the actin, however they were not moving, so we need to try again. Although our extraction techniques have improved, we now know that we need to keep our samples on ice more carefully and take care to avoid contamination. We are also running another set next month, so we will also use turkey and pool our samples so that hopefully we have sufficient quantity and quality to record contraction velocity results.
Okay. To start looking for differences in the protein sequences of the isoforms, we had some training in bioinformatics. We used the NCBI website to search for the myosin protein sequences by the name of the species and by protein sequence using BLAST, the basic local alignment search tool. The sequences found are shown here. We used Cluster Omega, a program for generating multiple sequence alignments of proteins, to identify differences between proteins. Cluster Omega can also be used to produce a phylogenetic tree to demonstrate how closely the species are related. The myosin tree fit with expectations as to the evolutionary history of the species we investigated, with ostrich being the most distantly related species in the evolutionary terms. We have realised how important it is to follow the protocols with a high degree of precision, and we have needed to keep refining the process. We will continue to search for further sources of bird cardiac material for a variety of species of different masses and heart rates. For our next session, we are also going to be carrying out ATP hydrolysis assays and using actin bound to magnetic beads. Adding to the myosin solution should pull the beads and myosin along with the magnet. If ATP is then added, the myosin will release from the beads and stay in solution. This will help us see whether our myosin is functional. If so, we can try again with the contraction velocity measurements at the university. We would like to finish by thanking all those involved in supporting our project, to the Wellcome Trust for funding to get us started, to Iris for the symposium, to Dr. Colthurst for his support, and to the University of Kent team who have given up so much of their time to train us in the techniques and guide us through the direction the project should take. And finally, to the Worshipful Company of Poulters for their interest in the project, sponsorship and offers of practical help in sourcing cardiac tissue.
Thank you. First question, did you find the penguin? If you go back a slide, there was a missing penguin. Anyway, do you have any questions from the students first? Okay, I have a question while you're pondering one. You said that it was interesting to do this research on birds, because there's a lot of research normally done on humans, but of course this was kind of applicable to school environment. Does it make you interested in going back in the future and looking at humans, or are you now kind of hooked on kind of bird physiology and want to go down that path?
It would be really interesting to look at humans. Obviously we can't because we're at school. But we want to look at as many birds as possible first, so we want to compare them all together and see how they look and stuff.
Mm-hmm. Thanks. No, I meant in the future, blimey, not at school. Crikey. Do we have any questions? Anyone got one about the whole process?
Can I ask?
Yeah. Oh, okay. Becky first.
Do you do quite a lot? I know your academic is here from Kent, and do you do quite a lot at the university as well as in the school? Because you seem to be doing lots of collaboration, which is fantastic. I just wondered how that works, because I know a number of projects just mostly do it at school. And are you getting turkeys ready for Christmas? No, somebody has to say that. So, no, about how you link with the University of Kent.
Who else wants to pass over to you again. Let's go for it.
Yeah, I haven't personally been up to the uni, but I know that a lot of our students have been up multiple times up to the uni to help test results and to learn about the processes up there. But yeah, they come down to us quite a bit and we go up there, so yeah, it's good.
It's so impressive for an outsider to see the amount of that collaboration between university and schools, and the amount of time that's given, by the university staff to the school projects is really lovely to see. Right, there was a question. Oh, with who was waving in the middle? Yeah, brilliant. Let's take this one in the middle. Thank you.
Hi. You were looking at contraction velocity in vitro, and I was wondering if you could tell me a bit about how that compares to what you'd expect to see in living cells.
Who wants to jump for that one? Don't all jump at once. Anyone give it a go? Give it a go, Ryan. We passed on that one. Great question. Chat to all the posters. Any more? Yeah, I'll run to you because you're at the front.
I'm assuming acquiring kind of tissue is hard in university. How does that limit your research, and in what way?
So obviously we are a school, so we're not allowed to actually have a lot of like birds and that. But I think like every 2 months, I think it is, we get like there's a chicken and quail and all that. But we try to get some more ostrich. We just recently got some of that, but there's obviously a limited supply because you don't just kill ostriches just for research. So yeah, it's hard to get it, but I guess the university allows us to do that. So.
Thanks so much. Yeah, over to you.
Were there any ethical concerns during your research? The fact that you're using a higher sort of animal as your research tissue?
Any ethical concerns?
the only ethics concerned is like where the birds have come from. So our new sponsor, which is basically a turkey farm, from what I know, they're obviously killed for meat, so it's sort of ethical unless you're a vegetarian.
No, birds were deliberately harmed in the making of this research. I'll come to you.
Is it more important for you to like do multiple repeats on, like, say, a chicken, or is it more important for kind of your research to do more rarer birds and widespread varieties of birds and species?
This is a workout, isn't it?
It's probably more important to do different birds so we get a wider range of results so we can compare from different species.
They're discussing this amongst themselves. It's planning the next step.
All right, should we give them a huge round of applause? Thank you very, very much.